All right, today we're diving into a secret that's absolutely core to our existence. It's about a component so ridiculously small it was hidden for almost all of human history, but it's the very thing that makes life, well, life. We are talking about the cell.
So let's just start with a huge question. If you had to pick one single secret ingredient that every living thing on this planet has in common, what would you guess it is?
It's not some huge, epic force you'd find in the oceans or mountains. No, the answer is much, much smaller. So small, you can't even see it.
And there it is, the cell. This is the absolute bedrock of all biology. Seriously, every living organism you have ever seen, and billions you haven't, are all built from these tiny units. Think of them as the essential Lego bricks of life. Okay, so what does it really mean to be a building block? Let's unpack how these microscopic cells manage to construct the incredible variety of life we see all around us.
You know, some organisms, like bacteria or yeast, are perfectly content just being a single cell. That's the whole show for them. But for more complex life, like plants, fish, or us, cells have to team up. They group together to form tissues, and those tissues build organs like your heart or your brain. And then those organs work together in systems. But the key thing to remember is that it all, every single time, starts with the cell.
Okay, but this brings up a really interesting question, right? If every living thing is made of trillions of these cells, how come we only found out about them a few hundred years ago? Well, the answer actually lies in the limits of our own eyes.
It all boils down to this one number, 0.1 millimeters. That's about how thick a single human hair is. And for us, that number represents a fundamental barrier. It's a wall our eyes just can't see past on their own.
It's something called the limit of resolution. So imagine you've got two tiny dots on a page. As you push them closer and closer together, you'll hit a point where your eye can't tell them apart anymore. They just blur into one single dot. For our eyes, that blurring point is 0.1 millimeters.
And this is exactly why we can see something like a fish egg without any help. It's bigger than our 0.1 millimeter limit. But most plant and animal cells, and definitely bacteria, they are way, way smaller. For thousands of years, there was this entire world living and breathing right under our noses, and we were completely oblivious. So, how did we finally break through that wall? Well, as it so often happens in science, it took a brand new piece of technology to pull back the curtain on a universe we never even knew was there.
I mean, just think about that for a second. For all of human history, this whole microscopic world was a total mystery. And then, in 1 year, 1665, everything changed. This is the moment it all happened. A scientist, Robert Hooke, was looking at a super thin slice of cork, and he saw, well, he described it as being full of these tiny, perforated, porous boxes. They reminded him of a honeycomb, or of the small rooms, the cells, that monks lived in. And, just like that, the word cell was born.
But here's what's so incredible. The tool that made this possible, Hooke didn't just stumble upon it. He built the machine himself. His very own microscope was the key that finally unlocked this hidden world.
So, finding cells was one thing, but how do you actually work with something that tiny? How can you possibly measure an object you can't even see? It turns out the method is surprisingly clever.
Yeah, the technique is actually pretty simple and elegant. First, scientists measure the full width of what they can see through the microscope. Let's say it's 5,000 micrometers. Then, they just count how many cells can fit side by side across that distance. If 25 cells fit, you just divide the total width by the number of cells. Voila! You've just calculated the size of a single cell. It's this beautiful little bit of math that makes the invisible measurable. But you know how it is. Science never sits still. Robert Hooke's microscope kicked the door open. But since his time, we have built tools that let us see things with a kind of clarity he couldn't have even dreamed of. The tech has just evolved like crazy. A regular light microscope, the kind you might see in a high school lab, is amazing for seeing the whole cell. But an electron microscope? That's a whole different ballgame. Instead of light, it uses a beam of electrons, which lets us see the mind-blowing tiny details inside the cell, all the way down to the nanometer scale. And this journey is not even close to being over. Every single improvement we make to our tools, sharper resolution for more clarity, better contrast to tell things apart, and more powerful magnification as zoom in further, it keeps revealing new layers of complexity and wonder inside the world of the cell.
So, For the first time in human history, we can't just see life's most basic parts. We can understand them, measure them, even interact with them. And that leaves us with one last, huge, fascinating question. Now that we can finally see the bricks of life, what are we going to build?
Welcome to The Explainer. Today, we're going to solve a biological mystery that actually starts with a simple potato from your kitchen. We'll find out how a cell's border patrol really works, controlling everything that comes in and goes out. Okay, so let's dive right into this. We're going to start with an experiment you could literally do at home right now. All you need is a potato, some water, and a bit of salt. It sounds super basic, right? But the results? Well, they're surprisingly revealing. So the question is simple. What happens when you put a potato in salt water? Now, don't let that simplicity fool you. The answer to this is the key that unlocks one of the most fundamental processes of life. It's going to reveal the secret world of the cell's border patrol. All right, here's the setup. It's super straightforward. We take a potato, cut it in two. One piece goes into a beaker of plain old water. The other piece, it gets a nice long bath in a pretty salty solution. Then we just let them sit for about an hour and see what happens. And check out the results. The potato in the plain water actually gets bigger, it swells up and gains weight, but the one in the salt water does the complete opposite. It shrinks, gets all wrinkly, and loses weight. So, what on earth is going on here? Well, the answer to our salty potato mystery is all down to a tiny but incredibly powerful structure that you'll find in every single living cell. It's time to meet the cell's official gatekeeper. And that gatekeeper is the cell membrane. You can think of it like a very, very picky bouncer at the door of an exclusive club. It gets the final say on which molecules get a VIP pass to come inside and which ones get told to stay out. So how is this bouncer built? Well, it's not a solid wall at all. Scientists have a great name for it, the fluid mosaic model. Imagine two layers of fat molecules, what we call a lipid bilayer, and they're constantly flowing and shifting around. And then embedded in this flowing layer are all sorts of different proteins, arranged kind of like the tiles in a mosaic. Now, while that lipid bilayer forms the basic boundary, it's really these proteins that are the stars of the show. They act as the actual gates and channels. They're the real decision makers, actively helping specific substances get from one side to the other. And get this, the cell membrane is only 7 to 10 nanometers thick. To give you some perspective, that is over 10,000 times thinner than a single human hair. It's just mind-bogglingly thin, but it's got this incredible power. Okay, so we've met the gatekeeper, but how does all this explain our potato experiment? It all comes down to one absolutely critical process. And that process is called osmosis. Simply put, it's just the movement of water across a membrane to try and balance things out. Water naturally flows from a place where there's a lot of water to a place where there's less of it. This isn't just some dusty biology class concept. It's how we preserve food by salting it. And it's exactly why IV drips in hospitals have to be so precisely balanced with our blood. This table right here is the perfect cheat sheet for understanding how this works. If a cell is in a hypotonic solution, which just means there's less stuff like salt dissolved outside the cell, water is going to rush in. If it's in a hypertonic solution with more stuff outside, water rushes out. And in an isotonic solution, everything's balanced, so there's no major change. And there it is, mystery solved. The potato piece in the plain water was in a hypotonic solution, so its cells swelled up with all that incoming water. The one in the salty water was in a hypertonic solution, causing water to flee from the potato cells, and that's what made it shrink. It was all about osmosis. But hold on, the story doesn't end there. What happens when we zoom in even closer to individual cells? And what if one type of cell has a secret weapon that the other one doesn't? So, when we put both plant and animal cells into a concentrated solution, something different happens. The animal cell does what we'd expect. It shrivels up completely. But the plant cell is weird. Its insides pull away from the edge, but the outer shape stays totally rigid. So, why doesn't it collapse? The reason for this strange behavior is a key difference maker between these two cells. It's an extra layer of protection, an extra piece of armor. Let me introduce you to the cell wall. This is the plant's armor. It's this tough, rigid layer that sits outside the cell membrane. And here's the key. Animal cells simply do not have one. This thing is all about providing structural support and protection. You can think of this wall as the plant's skeleton. It's what gives plants their structure. It's what allows a giant redwood to stand hundreds of feet tall against gravity and wind. It's mostly made of a super tough carbohydrate called cellulose, which is the most abundant organic polymer on Earth. And this stuff provides incredible strength. It keeps the plant cell from bursting when it takes on too much water. That's what makes vegetables crisp, and it keeps it from collapsing when it loses water. So understanding the membrane and the wall isn't just a fun science fact. It fundamentally explains why plants and animals are so incredibly different. It's a story of rigidity versus flexibility. And this really highlights a core principle in all of biology. Structure determines function. Plants are stationary, so they need that rigid support of a cell wall to stay upright. Animals, on the other hand, we need to move around, so our cells have to be flexible, and that's only possible because we don't have that rigid wall. As this quote explains, our ability to walk, to run, to move our muscles at all, it's completely thanks to the fact that our cells lack that rigid outer wall. Our flexibility truly starts at the cellular level. It's pretty amazing when you think about it. And that brings us to one last thought experiment. Just imagine for a second, what would happen to a plant, say a giant oak tree, if its cell walls suddenly became as flexible as our cell membranes? What would that even look like? Contemplating that question really gets to the very heart of why these microscopic boundaries are so incredibly critical for all life on Earth.
All right, let's dive in. We're about to shrink down like way, way down to tour one of the most incredible, intricate factories you could ever imagine. And the craziest part? There are trillions of them operating inside you right this second. We're talking about the living cell.
So here's a question for you. What do you, a giant redwood tree, and a single tiny bacterium all have in common? I know, it sounds like the setup to a really weird joke, right? But the answer gets to the very heart of what we're exploring today, the fundamental shared building blocks of all life on Earth. So here's our itinerary for this microscopic tour. First, we'll look at the two basic factory designs, the two main kinds of cells. Then we'll head straight to the corner office, the nucleus. After that, we'll see the assembly line in action, check in with the power and cleanup crews, and then we'll finish up by looking at some specialized departments you'll only find in plant cells. Let's get started. All right, first stop. When you boil it all down, every living thing on this planet is made from one of just two basic designs, or as we're calling them, two main factory models. Getting a handle on these two is really the key to understanding everything else. First up is the prokaryotic cell. The best way to picture this is like a simple one-room workshop. It's incredibly efficient, for sure, but everything's just kind of floating around together in one big open space. The tools, the instructions, the raw materials, it's all there. Its genetic blueprint, the DNA, isn't even in a separate office. It's just bundled up in a corner in a region we call the nucleoid. And now, let's meet the star of our tour, the eukaryotic cell. If the prokaryote was a simple workshop, this is a sprawling high-tech factory complex. It is all about organization, with dozens of separate rooms or departments, what we call organelles. Each one has a very specific job, and this compartmentalization is what allows for all the incredible complexity we see in life. You, me, plants, fungi, we are all built out of these amazing factories. So when you put them side by side, the difference is just night and day. It really comes down to organization and scale. The eukaryotic cell has its main office, the nucleus, and all its little departments neatly sealed off behind membranes. The prokaryotic cell is much more of a free-for-all. And just look at that size difference. Eukaryotic cells can be 10 times bigger, giving them all the room they need for the incredible internal machinery we're about to explore.
Okay, so with a factory this big and this complex, you better believe it needs a control center, right? A main office to run the whole show. Well, that's exactly where we're headed. Let's step inside the cell's headquarters, the nucleus.
You really can't overstate the importance of the nucleus. It's the brain of the operation, the command center, and the high-security vault all rolled into one. This is where the cell keeps the master blueprints for every single thing it will ever build or do.
So, if we peek inside, we see the DNA, our genetic code, all neatly packaged into these structures called chromosomes. To protect these priceless instructions, the whole nucleus is wrapped in a double membrane that acts like a fortress wall, with little pores that are like security gates, controlling who and what gets in or out. And that dark spot in the middle, that's the nucleolus. It's like a factory inside the main office that builds the very machines that will read the blueprints. How cool is that?
Okay, so we've seen the blueprints in the head office.
Now for the really exciting part. Let's head out onto the main factory floor and watch how those plans get turned into actual, finished products. Welcome to the cellular assembly line.
Now, this isn't just one machine doing one thing. Oh no. What you're about to witness is more like a perfectly choreographed ballet, a multi-step process with several key departments all working in absolute perfect synchronization. It's a true masterpiece of biological engineering.
Okay, so let's follow a product down the line. It starts when a ribosome, our little factory worker, reads a copy of the instructions. Then it starts building the protein, often right on the surface of the rough ER, which helps fold and modify it correctly. From there, it gets sent to the Golgi apparatus. Think of this as the factory's shipping department. It sorts, packages, and puts a shipping label on the protein. Finally, little transport bubbles called vesicles pinch off and deliver the finished product exactly where it needs to go. It's just amazing.
Now, an assembly line that sophisticated doesn't just run on its own. It needs a massive amount of energy, and like any factory, it's going to produce some waste. So for our next stop, we're visiting the essential utilities, the power plants and the cleanup crews that keep this whole operation humming along.
So let's meet the legends themselves, the mitochondria. You've probably heard the phrase, the powerhouse of the cell, a million times. Well, it is absolutely 100% true. No exaggeration. Without these little guys, the entire factory would grind to a halt in an instant. So how do they do it? Well, they take in fuel, like glucose from the food you eat. And through a process called cellular respiration, they break it down to release a ton of energy. They then store that energy in a special little molecule called ATP. You can think of ATP as the universal currency of the cell. It's the cash that pays for every single activity. And all those folds on the inside membrane? That's just to create more surface area so they can crank out even more ATP. It's all about efficiency. And now let's talk about the unsung heroes of the cell, the lysosomes. Every busy factory produces junk, broken parts, leftover materials, you name it. Well, the lysosomes are the cell's dedicated recycling and waste disposal team, and they are absolutely indispensable. These little organelles are basically just sacs filled with powerful digestive enzymes. They roam around the cell like little Pac-Men, gobbling up anything that needs to go. Old, worn-out machinery, bits of waste, even invading viruses or bacteria. But here's the brilliant part. They don't just destroy things. They break everything down into its basic components and recycle them, returning the raw materials to the cell so they can be used again. Nothing goes to waste.
So far, most of what we've seen you can find in an animal cell. But now we're going to look at a few specialized upgrades, some extra departments you'll only find in plant cells. This is what gives them their unique edge.
And first up is a real game changer, the chloroplast. We just saw how mitochondria are like power plants that have to burn fuel. Well, chloroplasts are like having your very own solar power plant built right inside the factory. They allow the plant to make its own fuel right from sunlight. This, of course, is where photosynthesis happens. They use that famous green pigment, chlorophyll, to capture the sun's energy and use it to turn carbon dioxide and water into sugars. Pure fuel. And here's a mind-bending fact for you. Just like mitochondria, chloroplasts have their own separate DNA. The theory is that billions of years ago, they were actually their own independent organisms. Next up is a structure that you literally cannot miss. It absolutely dominates the landscape of the plant cell. We're talking about the central vacuole. It's part massive warehouse and part internal skeleton. I mean, this thing is enormous. It's a huge, water-filled sac that can take up most of the cell's volume. Sure, it stores water, nutrients, and waste, but its most critical job is maintaining internal pressure. By pushing against the cell wall, it keeps the cell rigid and firm. You know when you see a plant wilting? That's because the central vacuoles in its cells have lost water, and that internal pressure has dropped. So we've talked about DNA as the cell's blueprints, but maybe a better analogy is software, the cell's operating system. In 2010, scientists did something that was pure science fiction. They built a bacterium's entire DNA code from scratch in a lab, inserted it into a different, empty cell, and watched as the cell booted up. It read the new software and came to life, running entirely on that new code. It was the ultimate proof that DNA is truly the software of life. And that incredible experiment brings us to our final, thought-provoking question. We've toured the factory. We've seen how the software runs the whole show. So if we, as humans, can now write that software, what does the future hold? What kind of new cellular factories or even new forms of life could we one day design and program? This tour of the cell, it turns out, isn't just a look at how life works. It's a glimpse into what's coming next.
All right, let's dive right in. Today, we're talking about something that's happening inside you right now, billions of times over. It's cell division. This is the incredible, hidden process that basically explains how we live, heal, and even exist in the first place. I mean, have you ever really thought about it? You get a nasty paper cut, and you're annoyed for a day or two, but then it's just gone. The skin is whole again. Or you get a haircut, and, a few weeks later you need another one. How does that work? It feels like magic, but it's not. It's the constant, amazing work of your cells.
And the sheer scale of this is, well, it's mind-blowing. Every single day, your body is estimated to replace hundreds of billions of your cells. To put that in perspective, that's roughly 1% of your entire body, recycled and renewed every 24 hours. And it's not spread out evenly either. The cells lining your gut might only last a few days, while your skin is in a constant state of renewal. It's this non-stop, absolutely essential maintenance program.
So here's the game plan for today. We're going to start with why our bodies are constantly rebuilding themselves. Then we'll take a peek at how scientists can actually watch this happen. From there, we'll get into the two main playbooks for cell division, mitosis and meiosis. And finally, we'll look at what happens when these incredibly precise systems make a mistake.
So where do all these billions of new cells come from? They don't just appear out of thin air, right? The answer is beautifully simple. Every new cell comes from a cell that's already there. It's a fundamental rule of biology, and it's the engine behind all life as we know it.
And this one process is juggling three gigantic jobs at all times. First up, growth. You started out as just one single cell, and through trillions and trillions of divisions, you became, well, you. Second, repair. That healing cut we just talked about, that's cell division in action, making new tissue. And third, reproduction, which is all about passing life on to the next generation. It is constantly working.
Okay, I know this all sounds a little abstract. It's happening on a microscopic level. So how on earth can we actually see it? Well, scientists have a classic trick up their sleeve, and it involves something you probably have in your kitchen, an onion. The root tips are growing so fast, they're the perfect place to watch cell division happen in real time.
And the method is actually pretty straightforward. You get an onion to grow some fresh roots, snip off the very tips, and then carefully prepare them on a microscope slide. The key part is adding a special stain that makes all the machinery inside the cell, especially the chromosomes, light up. Then you gently flatten it all out to get a nice, clear view.
And this is the aha moment. When you look through that microscope, you don't just see a bunch of identical cells. You see a snapshot, a process frozen in time. Some cells are just hanging out, some are gearing up to divide, and others are right in the middle of splitting in two. You are literally witnessing the different acts of the play all on one stage.
So let's get into the how. How does a cell actually divide? There are two main programs, and the first one is called mitosis. The easiest way to think about this? Mitosis is the body's perfect, high-fidelity copying machine. And the key word there is identical. In mitosis, a single-parent cell very carefully makes a full copy of all of its DNA, and then it splits right down the middle, creating two new daughter cells. Each new cell is a perfect clone of the one it came from. Same DNA, same number of chromosomes, the whole shebang.
And this process is the absolute workhorse of your body. It's how you grow. It's how your skin replaces itself every few weeks. It's how your body fixes a broken bone. For some simpler organisms, it's even how they reproduce. The bottom line for mitosis is this. It's all about making identical copies to keep things running and fix what's broken.
All right, on to the second master program, meiosis. Now, if mitosis is the copying machine, then meiosis is the creative mixer. Its goal is the complete opposite of mitosis. It's not about making clones, it's about creating genetic variety for sexual reproduction.
Now, meiosis is a really specialized process that only happens in the reproductive organs. It takes a parent cell and puts it through 2 rounds of division. But here's the critical difference. The final cells it produces, the sperm and eggs, or gametes, have only half the number of chromosomes as the original parent cell.
And this is why you might have your dad's eyes, but your mom's smile. Meiosis is designed to shuffle the genetic deck. It ensures that every single sperm and every single egg is genetically unique. That variation is the fuel for evolution, and it's why life on this planet is so incredibly diverse.
Okay, so let's put them side by side just to make it crystal clear. Mitosis is for growth and repair,
Meiosis is for sexual reproduction. Mitosis has one round of division, giving you two identical cells with the same number of chromosomes. Meiosis, it has two divisions, resulting in four genetically different cells, each with half the chromosomes. Two completely different jobs, two completely different outcomes.
Now, as you can imagine, both of these processes are unbelievably complex and tightly controlled by the cell. But what happens when that control system breaks down? What happens when an error slips through the cracks? Well, the consequences can be pretty serious.
If our copying machine, mitosis, makes a mistake and a cell loses its ability to stop dividing, that can lead to the uncontrolled growth that forms a tumor. When our creative mixer, meiosis, messes up, the consequences are different. An error there can lead to sperm or egg cells with the wrong number of chromosomes, which can result in genetic disorders, developmental issues, or problems with fertility. It just goes to show how much our health depends on these microscopic processes working perfectly.
So we end on this really fascinating idea, don't we? On one hand, life absolutely depends on the perfect, stable copying of mitosis just to maintain itself from one day to the next. But on the other hand, life also relies on the creative, chaotic shuffling of meiosis to adapt, to change, and to evolve over generations. One provides stability, the other provides variety. And it leaves you with a pretty big question to think about. For the long-term survival of life, which one matters more?
Let's crack the secret code that's hidden inside every single living thing. So what do a giant tree and you have in common? The answer connects all of us.
The answer? Cells. Yep, we're all made of these tiny, fundamental building blocks.
But you know, this was a huge mystery for centuries. Then science developed a unifying theory.
It started with plants, then moved to animals, and finally a game-changing new rule.
And all that work led to this, the cell theory. Basically, biology's official rulebook.
Okay, so those are the rules. But how do cells actually, well, behave?
Well, for one, they don't just grow forever. Their growth is actually super controlled.
And in animal cells, there's this really cool process for it called contact inhibition.
Right, so if they stop growing, did that mean they just live forever?
Nope, not at all. Cells actually have a natural, very organized way of dying.
It's called programmed cell death. Think of it like essential quality control for your body.
And it's absolutely crucial. It's how your fingers and toes were formed as an embryo. But what happens when this really elegant system just breaks down?
Well, sometimes that system fails. Abnormal cells can start dividing totally out of control.
You can see the difference right here. Normal cells are orderly. Cancer cells, pure chaos.
But it's not all bad news. While some cells break the rules, others have superpowers.
Way back in 1902, a botanist named Haberland had this absolutely wild idea.
This amazing ability is called totipotency. A single cell can become a whole new plant. So if one cell can do that, what other secrets are life's building blocks hiding?